Hi. So,
Good morning. And let us begin. Today, this is more, we're starting now, we're going to start to really hit more of the technical stuff rather than the historicals. This is the more of the anatomy classification part of the program, which you have to do. I mean, sometimes it's just, there's a little bit more memorizational component than anything else. So I have it, a lot of it written down, but the first thing is the notions that we have. What makes people live? Rule number one, made out of cells. Cell theory. All things come from cells and other, from pre-existing cells, except obviously the first thing that came about. They've got to have both RNA and DNA. Some kind of membrane, by implication, that means semi-permeable, gatekeeper, some stuff gets in, some stuff gets out. That's in there. Cell wall, we have one or doesn't have. You don't have cell walls, plants do. Okay, responds to the environment. Some ability to adapt to the environment. It doesn't have to be sufficient. It's not like you and I walking into a room and going, my chair is over there, and walking towards it. Okay, it's not that type of thing. Okay, it can be. Remember, we're dealing with microorganisms. There's not a whole lot going on. Their responses are very basic. If there's a change in an environment, there's some kind of chemical, or some kind of material, or if there's light, or if there's air, or if there's not air, there's going to be a change in the environment. But you're still responding. Yeah, and they obviously, if they're going to flourish, they have to reproduce. The notion of reproduction doesn't specify. You're going to see the reproduction here in bacterial and archaeal domains is very simplistic. They reproduce by something called binary, almost exclusively, by something called binary fission. It's really important. They just keep dividing in two constantly. So, and it's very, very simplistic. It's not, misconception number one is that everything is either mitosis or meiosis. That's much more sophisticated. And when it comes to eukaryote, even though that is not entirely true, there is both, typically eukaryotic organisms may do both, but certainly do typically maybe just mitosis. Because some of them are asexual. So the whole, we don't really have any type of quote unquote sexual reproduction in the eukaryotic domains, these simplistic. And archaea we're mentioning today in passing as well. And then lastly, energy transformations. What does that mean? Energy transformation is what we do. It depends on what we utilize for energy. Okay, if you're a plant, you utilize energy from sunlight and use carbon dioxide in the air and a little bit of water, and voila. All of a sudden you're making carbohydrates and giving off oxygen. You're doing photosynthesis. That's an energy transformation. What you and I do, we ingest organic materials. All that thing we were talking about at the end of last year, proteins and carbohydrates and blah, blah, blah. And that type of stuff. We ingest them, we break them apart because within their bonds, their chemical bonds, is the energy. So we break apart those bonds, we seize the energy because it takes energy to hold things together. We break them apart, releases it, and then we utilize that energy to do whatever we need to do. At the same time, when we're breaking that apart, a lot of times we use them rather than as an energy source, we're just using this building block or something. And if anything, we're going to put more energy into the system. So we're doing what are called energy transformations. A good word for that, metabolism. And inevitably with metabolism, you got to bring it in, you got to metabolize it, break it down or build it up, whatever you're going to do with it. And then finally, if there's refuse, if there's stuff we don't need, there's waste products, we get rid of it, whether it's for us, carbon dioxide and water and unused materials or materials that are of no benefit to us directly. That's about it. That's Living Things 101. It's not hard. Now we get to the differences that are here. And that's why I wrote it up this way more in a handout so you can have it. It's all based on something called the domain system. The domain system is founded in molecular biology. So we weren't able to analyze molecules as distinctly as we can today. Okay, we couldn't look closely at sequences of DNA or RNA. We couldn't assess their different types of characteristics. That started to, molecular biology kind of came to the fore maybe in the late 60s. And then by the time the early 70s rolled around, now it became a field unto itself. And I don't have the name written down. I always pronounce it wrong, probably. It's WOESE. In 1978, and I'm sure it was around before that, but that was the date of the publication, when Weiss, as I call him, or her, or maybe her, is published this treatise on the domain theory. It was based on two things. The density of the ribosome, and you'll see when we start talking about this, 70S ribosomes versus 80S ribosomes. S is just, it's a unit that called a Svedberg. It's a proper name, SVEDBERG units. And how you measure it, I don't know. But the fact that they're different in bacteria, let's say, than us, and that's really important, by the way. Okay. So he looked at the distinctions there, and then he looked at DNA sequences. And he found enough similarities in two of these things that he looked at, archaea and bacteria, he found enough differences to make them a domain unto themselves based on where they were located and some other factors. But other than that, there were enough similarities that he classified them, and he called them prokaryotic cells. Karyon is a kernel or a core, whatever word you want to use out of it. If you back in the day, if you looked in your biotext, and I'm sure they had it in AP1, they had like diagrams of chromosomes that were out, particularly if you're studying like trisomy, and you will, like trisomy 21 for Down syndrome. That was called a karyotype. That was, we couldn't do molecular analysis, but we could do physical analysis. We could basically map out the appearance of the chromosomes and see if a piece was missing, if there was an extra one or something like that. Okay, it was called karyotyping because we were looking at the chromosomes, kind of how it derives its word from there. So prokaryotic cells, pro or pre, early, and the implication was they were that way because they did not have a nucleus and other membrane-bound organelles. So that's the big thing here. Not just no nucleus. The nucleus is very specific. No membrane-bound organelles. And it tends to make things membrane-bound, and that's the highlight of eukaryote. Membrane-bound organelles. It implies that you have a degree of sophistication sufficient to really have almost little structures inside of a cell that are functioning independently, although linked together by a chain of membrane. Okay? So your ribosomes are doing business, your nucleus is doing business, your Golgi apparatus is doing it. It's all doing business by itself. Okay? But yet they're somehow chained together. So it's an increased degree of sophistication and an increased degree of complication. That is part of it. So there's a major difference. So he began to study these things and look at examples of all of it. So what he found was consistent with these two areas, both archaea and bacteria. If you want to say archaea, that's fine. They were all prokaryotic cells. They were very, very tiny. They only existed as a single cell unit. It didn't mean they couldn't do damage together, obviously, but effectively they were single cell units. They all had a cell wall. That's very significant. And indeed, we're going to spend an inordinately large amount of time in the next chapter talking about the distinctiveness of the cell walls, particularly of bacteria, because that's how we categorize them. Okay, and again, the name of it, I'll say it again, it's a proper name, Gram, capital Gram, for a Danish physician, and we classify them as gram positive and gram negative, and it is the most important test you'll ever do in that lab, by far. Okay. So they all have a cell wall. Their DNA is different than ours. Our DNA is in pairs. So we have 23 pairs of DNA. whatever, however you want to number them, and then we have an X and a Y, or X and an X, whatever we are, unless there's some unusual variation that's associated with it. Okay, so we've got all that straightened out. They have a single circle of DNA. You might remember me drawing it a little bit before. It's a circle of DNA. It's very, very large. Bacteria are probably about maybe a millionth of our size. Okay, they're really tiny, very, very small. Yet they have probably only, rather than having 1/1,000,000th of the genetic locations, they have about 1/10 of the genetic locations. So how do they manage to do this? As you will see in the next chapter, it's called supercoiling. It's like a rubber band that you keep twisting and twisting and twisting. That's what supercoiling is. You ever see one of those little airplanes with a propeller and the rubber band ringing a bell? The rubber band will kind of kink up. That's supercoiling. But it's very, very specific. We actually have an animation that I can show you about that. And that comes in the next chapter. And so they can compact this single circle of DNA. The DNA isn't any different in living things. Our DNA and their DNA still has A's, T's, G's, and C's. Their RNA still has uracil instead of thymine. Okay, so it's not a whole lot different. It's double-stranded DNA, just like ours is double-stranded DNA. Okay, so you have two strands that are basically, if you will, anti-parallel. One points up, one points down, except ours aren't just sitting there with open ends freely. They're all attached to one another. That's all. So it's double-stranded DNA. We'll confuse you with single-stranded DNA and double-stranded RNA, because RNA in us and living things is single-stranded when we get to viruses. More fun on that later. Okay, so they have a singular circular strand of double-stranded DNA, and it doesn't bear the name of nucleus. We call it the nucleoid, because it looks like the nucleus because it's all compacted and coiled and it's sitting in one place. It just doesn't have membrane around it. It's sitting here in the cytoplasm. The ribosomes have a different density, and this was part of his work, Weiss. He looked at the ribosomes. Ribosomes, if you remember from AP1, you have a large and a small subunit. Okay. The large subunit, you have to know this, is 50, the small one's 30 in 70S ribosomes. When they come together, the density changes. It only is 70. So it's like there's something happens when they combine, they become more dense or less dense, however you want to look at it. It changes the density. No membrane-bound organelles, including the nucleus. Everything is just free in the cytoplasm. Doesn't mean it doesn't move around. It doesn't mean there's not an infrastructure in there. For years, we didn't think, it was kind of like when they used to let you have in Pittsburgh, have plastic bags. And, you went to the grocery, you went to Giant Eagle, and you put everything in the bag like that. They used to think bacteria were like that. It's just like everything's thrown in. There's no, here, there's structure. We now know that. Okay. They replicate by binary fission. Super important because it's very uncomplicated and it's very fast. And that means they can get giant numbers very quickly. I mean, in a day, you're going to start with one, you could have 1,000,000 of them. It doesn't take long. It's one of those, we call it an exponential curve. We explore that in our chapter about basically a bit about energetics and structure and growth that are all part of that. Okay, so it's binary fission. Very uncomplicated process. I say it all the time. Most of their energy transformations, because they don't generate nearly as much energy as we do ordinarily, are dedicated to one thing: reproduction. And that's the bad news, because they reproduce so fast, the ones that hurt us can overwhelm us relatively quickly. Moreover, many times their waste products are the problems, because to us, they are toxins. They're foreign to us. And either we respond immunologically, or they're directly toxic to certain kinds of cells. Like we were talking the other time about tetanus toxin and Botox. Those poison nerve cells, for lack of a better way to describe it. A lot going on there. Okay? They respond to environmental changes just like all living things. Okay, look what we do. We talked about pasteurization. We're heating it. When you heat it, it changes its ability to do business. And if you heat it enough, the ones that are going to cause typical foodborne illness die. If you can hold it again at 140 degrees Fahrenheit for roughly 30 minutes, they're dying. Mostly, and most of the ones, and the ones we're going to talk about for the most part, derive their energy from organic compounds. And just the way I described before, just like we do. We are what are called organoheterotrophs. We eat different things that are made of organic chemicals. We break them down for energy, or we break them down for building blocks and reassemble. That's what you and I do. It doesn't matter whether you're vegan or vegetarian, omnivorous or herbivorous, you know, whether you're a cow or whether you're a dinosaur or whether you're an us. Doesn't matter. We're all basically doing business the same way. How are they distinctive? Bacteria, and this is this. When you use the term unique in science, unique doesn't mean, oh, it's really interesting. Unique doesn't mean it's really exciting. Unique doesn't just mean different. Unique means it's here and nowhere else. The only thing on the planet that's got this magic material called peptidoglycan, which is a combination of peptides, which are amino acid chains, and glycans, which are carbohydrates that have nitrogen in them. We'll get into all of that. That's where it gets its name, peptidoglycan or peptido. I always say peptido. The bacterial cell walls all have some amount of peptidoglycan. Not all the same, not all arranged the same. That's that whole gram-positive, gram-negative thing we're going to get into ad nauseam, and that is unique to bacteria. Additionally, they live everywhere. So they can live in normal climates, more or less like ours is. So there's plenty of bacteria, whether we had snow on the ground, it was freezing overnight, or we're going to have plenty of bacteria on a rainy day in the 40s like it is today. Doesn't matter, they're still here. They just, you know, maybe we'll have more of certain kinds depending on certain conditions, in the middle of the summer versus the middle of the winter, etc. That type of thing. So, and basically they can live in all sorts, even in extreme environments, they're just not confined to that area. And we're going to go through a whole classifications of the different environments they live in, mostly based on temperature. So, and the ones that bother us are the ones that, you know, thrive around our temperature, specifically body temperature. Good old 37 degrees Celsius, 98.6 for you fans. Okay, we got all that down. The peptide, when something is unique, think of it this way. Bacteria have it, we don't. That makes it a target. So if we can destroy peptidoglycan, we can kill the bacteria. And since whatever we're using that targets the peptidoglycan doesn't hurt us, more or less, it's a really great item to target. So if I play word association with you, and I say infection, when you go to the hospital or urgent care, what do they give you for an infection? What do you get a prescription for? It's a common word used every day. You have a bacterial infection. Oh my God, I have got green stuff coming out of my nose and blah, 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 blah. What do they give you? What does the doctor give you a prescription for? A generic name. Thank you. Did you all hear that? Antibiotic. Play word association again. If you say antibiotic, what's the name? What do they usually give you a prescription? What might they have given you a prescription for? If you weren't allergic to it, hint.
Penicillin.
Penicillin. What does penicillin target? It blocks the assembly of this neat molecule, peptidoglycan. But I told you the problem with penicillin, if you do not know, it is derived from a mold. You'll commonly find it on white bread that's getting moldy. So, don't tell me you haven't seen it. So, I know you. You see the refrigerator in the dorm room and stuff has been sitting there for, and you start to get that kind of blue-green kind of, you know what I'm talking about. That's penicillium. Doesn't take long. That's penicillium. So that's the problem with it. It's a mold or a fungus. And one of the things that mold and fungi produce in a lot of us are allergies. So the biggest single problem, and I'll say it to you all the time, is allergies. And the saddest words a doctor can hear are these three: allergic to penicillin. Because it's the best of the antibiotics, as long with ones that are in the same different category. We'll go into a long discussion about that. We're going to keep going, and I'll give you the next nugget. So we've got that. The ribosomes, it's got 70S ribosomes. That's pretty unusual. Okay, so if someone's allergic to penicillin, the other target we use is the 70S ribosome. Okay, so if does anybody know what typically the drug of choice is? That's an important term. It comes to therapeutics. It's not about addictive disease. That's a different term. Okay, same. It says drug of choice. Addictive disease drug of choice is a person's a heroin addict or a cocaine, whatever they are. That's their fentanyl, whatever they're using. Okay, as opposed to drug of choice in a medical field means it's the prescription you're supposed to write, and if you don't write it, you could have a very good reason for not writing. Because that's what lawyers ask. What's the drug of choice? Doctor, please tell me. Oh, the drug of choice for this is penicillin. And you didn't write a prescription for penicillin? Why was that, doctor? Because the patient was allergic. Okay, good answer. So what do you give somebody who's allergic to penicillin? Other prescription for antibiotics. Pardon me? Amoxicillin is similar to penicillin. It's in the same family. You may not be allergic to it necessarily, but it is in the same family. Normally you'd have to test first. Anybody else? Something works really fast, you hear about it.
To start with,
Z.
Zyrtec.
No, Zyrtec is a good answer. Zyrtec is an antihistamine, a non-drowsy antihistamine. I took one this morning.
Oh, I took one last night.
Oh, God. Well, I knew we were similar in those ways.
I knew it.
Cetrazine for both of us. Okay. You ever hear of a Z-Pack?
Yeah.
Happy dance. Z-Pack is something called erythromycin, except it's kind of, they played around with it and they made it so, they really didn't make it work faster. They made it stay in your system longer, and it became a very, very beneficial drug. Okay? Z-Packs and other members of that category target the 70S ribosome. The biggest problem with them is not allergies, it's side effects. When you're dealing with antibiotics, the number one side effect problem people have is gastrointestinal. Vomiting, diarrhea, constipation, whatever you want to call it, heartburn, anything. All of those can come. Why there are side effects is it's not unique. As you will learn shortly, in our cells, inside of our mitochondria, we have 70S ribosomes. And so even though it's targeting certainly the vast majority of the bacterial ribosomes, unfortunately some of ours get in the way. So that's a little, and that is a microcosm of why we study things in such detail. Because frankly, no one's going to ask you, particularly if you're a nurse, oh, does that target 70S ribosomes and what density unit it is? No. Is it going to help me? Yes, they're going to ask you that. Okay, but it's important to know why a certain choice is. Oh, someone may say, why am I getting a Z-Pack and not the amoxicillin that I usually get for this? Oh, because we think you might be allergic to that. That's fine. I get an answer. People just want clarification. They're not going to ask you a whole bunch about that stuff. Okay, so we've got the 70S ribosome. They replicate by binary fission. Organics, we covered all this. Bacteria have the cell wall. They live everywhere. Archaea is the only time we were talking about it. Other than that, there's a test question. They're confined to extreme environments: polar ice caps, volcanic caldera, bottom of the sea, salt flats. Pick it. Someplace where it's an environment that you would not be able to thrive in. Interestingly enough, they have a cell wall, but it does not have peptidoglycan. They gave it a name, pseudopeptidoglycan, because it's not quite the same. And because it's in extreme environments, there's some interesting alterations in its cell membrane that you certainly don't have to know about. What's really unique about that, if you remember where life began on this planet, in extraordinarily toxic environment, gaseous, hot, irregular, just awful. Okay. Interestingly, when they sequence DNA to do this theory of domains, there are more similar chains of archaeal DNA in our DNA than in bacteria. They're studying that. What does it mean anything? Maybe not. Is it coincidental? Possible. But I mean, originally our cells started in an extreme environment. That maybe gives some credence to that idea. Just saying. And again, I'm not an expert in this science. So that's why, and that's really about as much because we don't deal with them very much. I mean, unless you're involved in a field that goes to these extraordinary areas, we deal with some oddballs, but not these. And so we're going to, and again, we'll look at it more closely. That's the first part. So by and large, our course focuses a ton on bacteria, none on archaea, and this is the basis. We also focus a ton on eukarya, particularly protozoans and fungi in that family. So they include us, animals, and they include plant cells, trees, bushes, whatever. Those are macroscopic, so they're not microbes, so we don't bother with them. They aren't limited to one cell. They have to be made out of cells, but they can be single or multicellular. So, and even some microbial forms, certain kinds of yeasts and mold are still in the category of microbial because we can't see them. When they kind of accumulate enough together and become, say, a mushroom, now they're macroscopic and now they're out of the discussion. Although, again, very tasty. Okay. Not throwing a food nugget here. They don't have to have a cell wall. They have membrane-bound organelles. Remember, that's the distinction. Pairs of DNA, also double-stranded. Okay, and the organisms differ. So an elephant has so many, and a mouse has so many, and a fruit fly has so many, and we have so many. Okay, so they're all different. I don't know the numbers. My colleague, Dr. Hannah, is our resident zoologist, and so she could probably tell you better than I can. Okay, they produce mostly, they have ADS ribosomes. That's a big difference. The ribosomes, other than the ones that are found inside mitochondria, those free and bound ribosomes you learned about in AP1, exciting, isn't that? Those are ADS ribosomes, so they're larger and have a greater degree of density. Okay, most of the reproduction is mitosis or meiosis. There should be an E in there, and I pardon for the misspelling. And there's also some oddball budding and spore formation. To be fair, spore formation is something that's a little bit different than endospore formation. When you get into lab, and if they're having you look at fungal things, typically pre-made slides of fungi, you'll look at different spore formations. Membrane-bound organelles live all over the place. This is important. Basically, we have the yeast family, a lot of impact on us. Yeast, because of all it does for us, and yeast infections are a big player. Fungi, a tremendous amount of skin problems start with fungal infections. Not athletes would truly be the most common of all of those. Okay, are singled or multicellular, and they're interesting because they have cell walls, but unlike plants, they don't have chlorophyll. So the fungi are sort of, they're mostly land-based to start with, okay, and they are much more, let's say, very, very similar in their construction to plants, but certainly not the same. They're typically on land, and we call them decomposers or saprobes. That's a fancy name, and bacteria do some of the same thing. So when you see roadkill on the side of the road that hasn't been taken care of and it starts to deteriorate, fungi, you see them all the time. If you go out, if you like hunt for mushrooms, okay, if you're into that type of thing, and we're not there. In about April, you start looking for things called morels, which are and we have a very active in Allegheny County, the Mushroom Society. And my wife and I have gone on a few of those mushroom hunts. It's just a lot of walking outside. It's cold and wet. But where you're looking on the ground, and you're looking on trees and on certain kinds of trees.
for those kinds of mushrooms. So you go foraging effectively. And you ought to, and if you want to do that, know what the heck you're doing. Just don't start eating it because it looks good. Because you could be in big trouble. Go with somebody who knows what they're about with regard to that. So we talk about that a lot. They're a fancy name that we associate them. They're called exodigesters. You and I, we eat something, enzymes in our gut break that stuff down so we can absorb it. So we've already put it inside our alimentary canal. Saprobes and fungi, they secrete or give off chemicals that break it down. Once they break it down, they gobble it up. That's what's meant by an exodigester. Not a big deal for you, but. And now the other category that's really significant, protists. Protists do not have a cell wall. There are two in the family. One is called algae. We talked about that before with regard to oxygen. Algae, by definition, have chlorophyll. They are the only eukaryotic microbe, so it is unique to algae. Some bacteria have pigments in them, but they're not eukaryotic microbes. And then protozoans, all those things you saw that illustration of, the things you looked in the pond water, even though there's some dirt and stuff that appear that they might have pigment, and probably because they've absorbed some materials that were pigmented, it isn't included with the package. So algae are like plants. They have chlorophyll, they have cell walls, and if we can see them, we start to see them, they start to become multicellular. Photosynthetic, by definition, only water-based. We're protist, single cell, no cell walls. They're like little single-celled us. No chlorophyll, can live in land or thrive in water. Again, I don't do the worms. So again, when it comes to disease, statistically, the second most common sexually transmitted disease is carried by a land-based protozoan organism. Okay, that land-based, I should have put the word protozoan in there. It's called Trichomonas vaginalis. It is extraordinarily common. The big three when it comes to frequency of sexual, and we go over this when we do that unit at the end of the class. HPV, human papillomavirus, Trichomonas vaginalis, herpes, genital herpes. Those are the big three. When it comes to number of cases based on number of new case presentations per year. Just alone with that. And most of the statistics will bear that, certainly. They typically put them in that order. So it's not inconsequential. And you remember I mentioned malaria, the largest killer in equatorial Africa, it's protozoan. So they are significant. Okay? If you have, I think I told you about it, if you have well water, the type of thing that makes people sick and they get all kinds of, they get giardiasis or what's called amoebic dysentery, protozoan. So there's plenty of activity with regard to that. Which brings us to the last members of the microbial world that are oddballs. Okay, and they are and the fact that we describe them as acellular forms means by definition they're not living. And I will make the same thing. I'll say, how do we kill this virus? You can't. It's not alive to kill it. So it's better we either look at active or inert. It's a better way to describe them. But colloquially, we say that all the time. You'll see, go into the labs, go into any one of those areas and you see those little pump. Oh, it says virucidal. That means it kills viruses. But understand when you market it to people or to universities, we want to see that stuff. It makes us feel better. There's a lot, we'll get to that. There's a lot of misnomer things. It's like when you go into Giant Eagle and they have Clorox wipes. There's no Clorox in those wipes. Are you kidding me? But they know the name Clorox makes sense to me. Oh, it's Clorox wipes. I love it. In the grocery, you go to Giant Eagle, you go to Walmart. Oh, they got great. Really? If it makes you feel better, go ahead and do it. It doesn't do a whole lot. Let's be clear. You know why? Okay. Because where's the first place? You go to Giant Eagle, the first place you usually go right into is fruits and vegetables. If they put Clorox in those wipes, and you had it on your hands from wiping them down, and you grabbed, the first apple or pear or peach that you saw, and you took a bite out of it when you got home and go, ugh. Oh, because it's beyond there. They put something that doesn't have that sort of residual flavor component to it. They don't. Don't let them fool you. Be a label reader. Don't believe them until you see what's on the label. Turn the back of the thing, like that. Right. So they're non-living, but they live inside cells. So viruses, and we spend a lot of time on viruses. Viruses are what are called obligate, meaning they have to, intracellular, inside of the cell, parasites. They live and thrive once they're inside of our cells. That's the way they work. Okay? And they damage them by residing there, permanently damaging or destroying the cells, and very, very famously by generating, because they're foreign, a profound immunological response. You're going to see that's a big distinction between bacteria and viruses. Bacteria make toxins, they make poisons. Sometimes your immune system gets involved in creating the damage, an overly exuberant immune response. Viruses, by and large, if you have a flu or a cold, that's an immunological reaction. It's not viral destruction, if you will. Okay? Viruses have DNA or RNA, never both. So the first way we classify them, are they a DNA or an RNA virus? We classify them genetically by their genome. Again, we have some oddballs. In living things, DNA is double-stranded, RNA is single-stranded. We can have the reverse of that in viruses. I don't make a big deal of that because it's not that important. For instance, you'll see that when we get to the viral unit, single-stranded, negative sense, positive sense. I don't get into that. I really don't think it's necessary to read in that depth. At the very limit, you want to think of the simplistic form of a virus, it's got either DNA or RNA, and at the very least, it's surrounded by a shell of protein. That's what it is, a minimum. To this day, I can remember when we did virology back in 1974, when I was in medical school, and the lead virologist for the Commonwealth of Pennsylvania, my instructor, he came in and taught our virology course for a friend of his who was our main professor who was there. He went up and he drew this. And he said, that's what we know about viruses. Okay, 50 years ago, I'll buy that. He said, and it basically is drawing rabies. It's got genetic material in there, and it typically has more or less a bullet shape, because rabies is a pretty good sized virus. And you can see it microscopically, some age. And like this, and he said, most of the time that's what we do. And then we began to talk about viruses. Very, very interesting. I'll have another story about him down the road in this class. He was the lead guy for the Commonwealth of Pennsylvania. So, had a protein shell at the very least. We know now that they may or may not have a membranous envelope. Very much a membrane like a cell does. A phospholipid bilayer. Not the same, but similar. Okay, and so another way that we classify them is, are they enveloped? Do they have a membranous envelope? And colloquially, we call them naked viruses if they do not. And by definition, they can infect all living cells, including bacteria can get a virus like you can, archaea can get a virus, and obviously we can get a virus. And it's become much more important because, like a lot of viruses, it damages the cell. So as we run out of antibiotics, we're starting to look at viruses that affect bacteria, since they don't affect our cells, as alternatives to antibiotics. You learn about them, they're called phage, Phage, or bacteriophage viruses. And then lastly, on this little saga we're going through of the basic anatomical construct, are prions. Proteinaceous infectious particles is really what they are. No DNA, no RNA. They're misfolded proteins that, for all intents and purposes, basically bang into other proteins and cause them to misfold. And proteins make up a lot of structures, particularly in our nervous system. And so our knowledge about prions, albeit is comparatively limited, they cause these misfoldings to alter function, particularly in the nervous system and And we'll talk about that a little bit when we talk a little bit about mad cow disease, how it's spread, and things that are similar to that. I mean, for all we know, it may all be the same prion. We think by and large it comes from ingesting material. It's way more something we eat, and over time produces the problem. So it's very interesting about that, and there's a lot of information about it. So that handout's there. You can see additional chapter 1 micro. It's got that little parentheses 2, and it's right on the blackboard. So have that. You have the historical figures. You have the stuff with the resurgent and emergent diseases, all on handouts. Because I was doing all that typing when we were doing that other stuff. I gotta make sure that this is not the right one. I want to go back and see the end of our PowerPoint. Bacterial shapes and arrangement, fundamental characteristics of cells. Is this it? This is chapter 1. So I got it right. So again, single cell versus immense numbers of cells. When we start looking at the anatomy, we do a lot of characterization about shapes, and shapes also about how they cluster together, if they cluster or chain together. The word staphylococcus. Coccus is a spherical bacteria. When it comes together and forms a cluster is staphylo. That's where it gets it. Strepto means chain. So it's a strep throat has nothing to do with any pattern in your throat. It's the way the bacteria appear microscopically. And you will see that in the laboratory when you start to stain these things. Okay? They contain some type of plasm in a cell membrane, chromosomes with DNA, ribosomes, complex function. The eukarya organelles, this is just reiterating what we did. Bacteria, no nucleus, or not other organelles, other membrane-bound organelles. There's not a whole lot in there, but there's certainly ribosomes. So that's organelle. That's an organelle by any definition. So PowerPoints have mistakes. Okay. So we classify things. This is Linnaean taxonomy or nomenclature. And thankfully, you probably did this in high school where you had vertebrates and invertebrates and phyla. Am I ringing a bell? Notochords. You see, you had that. I see that. Okay. All those, why would, yeah, I remember my health teacher who was a gym teacher. You know how you can tell the difference between a gym teacher and in my day and age and the janitor? Because they both had the khaki pants and the shirt, but it was a whistle. Because the gym teacher had the lanyard and the whistle. You all had that gym teacher, man or woman gym teacher. Listen up. Didn't they ever tell you to listen up? Well, listen up, everybody. And I'd like to do this. You go, Stein, what are you doing? I said, I'm listening up. Do you want me to listen down or listen straight ahead? Take a lap. Where should I take it? Out for dinner, perhaps? Take another lap. Oh, we're having company for dinner? I was that kid.
Did you get in trouble a lot?
Oh, I was a commuter to the principal's office. I had my own little chair in detention, like a director's chair with your name on it.
Nice. They must have really liked you.
I was that kid. One in the front of the class. Teachers, are you ever sick? No, it would make your life too easy. Now I'm the teacher. See?
Tables have turned.
Goes around, comes around. See? I can't turn one of these tables. I could turn a chair around. Stop. So what's easy for you is you don't have to do any of that crap. Okay? They have all of that for living things. We just do, and I did it before a little bit, genus and species. So when you talk about E. coli, why do we call it E. coli? Well, coli is where we find it in the colon. It was named after the doctor who identified it. His name was Escherichia. Doctors are vain. He named it Escherichia coli. So we just call it E. coli. So Staphylococcus aureus, like I said before, we just write S aureus. That's perfectly acceptable anywhere on a test, and it's also acceptable on a medical chart, which for some of you have to be aware of. You only use acceptable abbreviations, because if you don't, lawyers love that stuff. They're just looking to blank you over on that stuff. Yeah. Fornicate you over. I was being good. Sorry. So there you are, E. coli. And by convention, you should either, you don't have to do it for me, italicize or underline them. And chart, I would underline. Okay, and here's the system. No, we're just doing, genus is the first name, capitalized. First letter, species, the second name, like we're Homo sapiens. Capital H for that, sapiens is the smallest route. And there we are. And you can go through all that nonsense. And I'm not doing the evolution. And it's probably true. This is the idea of what we call a novel virus. COVID doesn't mean, it means coronavirus identified number 19. And we'll talk, when we do the viral unit, we'll talk about coronavirus is nothing under the sun, by and large. Okay, but understand that when we talk about a novel virus, it's, again, it has somehow incorporated some type of genetic material from a new species or new combination of species that we have not become acquainted with. And it produces or provokes a rather extreme immunological response. And we're not doing that, those different models. This is the Weiss system, that's what I was talking about. That's the ones we use, and that's everything. So we're done with chapter 1. We will begin, I think it's chapter 3, which is really sort of the anatomy, more the anatomy than the physiology of bacteria. Next time we meet. So see everybody on a Friday. Thank you very much.